My dear students, welcome back to Plus 2 Biology from the chapter Sexual Reproduction in Flowering Plants. Our today's topic is Megasporogenesis. In the last session, we have discussed microsporogenesis and you have studied the structure of pollen grains too. Okay? And today, let's discuss the Megasporogenesis. Then you know the female reproductive word of a flower is gynoecium. Isn't it? The gynoecium is the female reproductive word of a flower. Then the segments of gynoecium, the individual parts, components of gynoecium are carpels. Okay, also known as pistils. And you have learned the plants can be with a single carpel. Flowers can be with one carpel, monocarpellary, or it can be with many carpels, polycarpellary. And if there are many carpels, a polycarpellary gynoecium can be either apocarpels, the different carpels, the number of carpels may remain fused. They may either remain free or remain fused. Okay, when a flower is having more than one carpels, these carpels may either remain fused. If they are fused, the condition is called syncarpus. Fused gynoecium. A gynoecium in which carpels are fused is called syncarpus. And whereas a gynoecium in which the carpels remain free, it's called apocarpus. And the textbook give a better picture of two examples. For an apocarpus gynoecium, it's Michelia, example given, and for a syncarpus gynoecium, it's given Poppy, Papavia somnifera. In your book, so many examples are there. These are your textbook direct examples. So, apocarpus and syncarpus gynoecium. I think it's clear. The female reproductive whorl of a flower is gynoecium, and in the flower, the female reproductive parts, the gynoecium has individual segments called carpels. Carpels can be also called as pistils. Okay, and mostly the flowers can be in both the types. The flowers can be of two types, either with single carpel or with many carpels. And if there is single carpel, it is called monocarpellary. If there are many carpels, more than one carpels, it's called bicarpellary. Then on polycarpellary. The condition is, if there are many carpels, polycarpellary gynoecium, it can be either syncarpus or an apocarpus. Syncarpus means carpels remain fused. Apocarpus means carpels remain free. Okay, I think you got me. The examples are for apocarpus gynoecium, one example is Michelia chambaka, Michelia. For syncarpus gynoecium, one example is Papavia somnifera. Okay, so gynoecium is made up of carpels. Then what is the structure of a carpel? About the structure of carpel. You have already learned the structure in last year +1 syllabus. Anyway, just to have a simple idea, a carpel is having stigma, style and ovary as the three parts of the carpel. Stigma, style and ovary. And here ovary is the basal bulged portion, style is the middle portion, and stigma is the upper expanded portion, which is the landing platform for pollen grains. The pollen grains have to be deposited here. Okay, and this is the landing space for the pollen grains. Style is the stalk in between, and ovary is the fertile lower basal region. And these are all fit to the thalamus. And the ovary consists of a specialized tissue in the center. Okay, maybe differently located. You have studied different types of placentation. Typically, when I speak, the ovary has a specialized tissue which is called placenta. Okay, the ovary, there is a special tissue called placenta, and attached to the placenta, we can see the ovary consists of, attached to the placenta, we can say the ovary consists of many or one or many ovules. Okay, so about the structure of carpel. Okay, one carpel has a basal bulged portion called ovary, then style and an upper landing space called stigma. Okay, and stigma, style and ovary, the three parts. Here in the ovary, stigma, style and ovary, the three parts. And ovary, there is a specialized tissue called placenta. Attached to the placenta, we can see what? Ovules. Okay, then keep in mind, the ovule is the female sporangium. You have learned anther, microsporangium. Here it is, ovary consists of the megasporangium. Anther consists of microsporangium, 4 microsporangia. Okay, here the ovary consists of 1 to many megasporangia. Maybe sometimes 1 megasporangium, or it may be more than 1 many megasporangia. So what is a megasporangium? This ovule is a megasporangium. So ovules are megasporangia. Singular ovule is megasporangium. Did you get me? Just anther consists of 4 microsporangia, whereas ovary consists of the basal bulge portion, ovary, the fertile portion, consists of 1 or many megasporangia. Okay, then come to the structure of a megasporangium or ovule. The megasporangium can be of different types based on the positioning and all. Anyway, typically an angiosperm ovule or megasporangium is called anatropous ovule. Okay, typically angiosperm ovule is anatropous ovule. An anatropous ovule is inverted ovule. What's meant by that we will discuss. It's an inverted ovule. An orthotropous ovule means it's a straight ovule, just for a comparison. And if the base is here in an orthotropous ovule, the apex is here. Base here, okay, apex, normal concept. Okay, whereas in an anatropous ovule it is inverted. The base is here because of the bending, apex also this side. Okay, inverted. Base and apex at the same side. Then it is called inverted. So anyway, a straight ovule is having at 2 ends, the base and the apex. Whereas here, the base and apex are at the same side. Anyway, we are coming to the technical terms. I will draw an anatropous ovule neat, then you can understand what is an anatropous ovule. The stalk of the ovule is called funicle and it has the body, okay, and it is surrounded by the protective coverings called integuments. Okay, the protective coverings are called integuments. Okay, so this is the stalk of the ovule, and I told you the stalk of the ovule is called funicle. Okay, this is the body of the ovule and the tip part. There is an opening called micropyle. The tip has come to downward because of the bending. Okay, so it is an inverted ovule. And the tip portion, there is an opening called micropyle. The opposite portion of the micropyle is called chalaza. Okay, the opposite end is called chalaza. And the 2 protective coverings are integuments. Okay, and like just to compare it with the microsporangium structure, 4-layered outer covering. Central tissue in the microsporangium was porogenous tissue. Here the central tissue is called nucellus. Okay, the central tissue is called the nucellus here. Now you to shade it, just have this idea. There is a central homogeneous mass of cells. This is called nucellus. Okay, did you get me? And the funicle is attaching to the body. Where the funicle attaches to the body, that space is called hilum. Hilum is the region of attachment of funicle to the body. The stalk attaches to the body of the megasporangium at hilum. Hilum is the region of joining. And this is the structure of an anatropous ovule. The stalk of the ovule is funicle and funicle attaches to the body at a space called the hilum. And the body of the ovule has the basal end called chalaza and the tip end called micropyle. And central tissue is called nucellus. It is surrounded by 2 layered integuments. Okay, 2 outer coverings. The integuments are incomplete. They are not there at the tip. So at the tip there is a space, an opening. That is called micropylar opening. That end is called micropyle and the opposite end is shalasa. Okay, did you get me? Then what is an inverted ovule? What is an anatropous ovule? Where the hilum and micropyle, the funicle and the micropyle comes at the same side because of the bending. That's why it's called inverted ovule. This is a common type among angiosperms. Okay, so we have discussed the structure of megasporangium. We have already discussed what is a carpel and what is a megasporangium. Okay, when we compare it with the male, what is instead of carpel? Instead of carpel it is stamen. Instead of megasporangium it is microsporangium. And the next one, instead of pollen grain, here it is embryo sac. The male gametophyte was pollen grain, whereas the female gametophyte is named as embryo sac. So what we have to learn is, next is embryo sac. In order to get into embryo sac, the process is megasporogenesis. You have already learned microsporogenesis. You have defined it better. So how can you define megasporogenesis? Where it takes place? It takes place inside the nucellus. Microsporogenesis took place inside the sporogenous tissue. Here, megasporogenesis takes place inside the nucellus. Okay? So what is megasporogenesis? Megasporogenesis is the process by which megaspores are formed from megaspore mother cell by meiosis. Did you get me? The same way you can define. Okay, you can define it. Megasporogenesis is the process of formation of megaspores by meiosis from megaspore mother cell. It takes place inside the ovule. Okay, so within the nucellus. Then let us see what happened at the megasporogenesis. So you know the central tissue is what? Nucellus. And within the nucellus, just compare, in sporogenous tissue, every cells act as the pollen mother cells. Whereas here in nucellus, mostly one cell, single cell near the micropyle function as megaspore mother cell. Not all the cell, only one cell. One cell at the micropylar region functions as megaspore mother cell. This is the megaspore mother cell. This cell undergoes meiosis first to form two cells. That is megaspore dyad. Similar case, earlier same case. It completes the meiosis second to form a linear tetrad. Earlier in microsporogenesis you have seen a tetrahedral tetrad. Here is a linear tetrad formed. So what is this? This is the tetrad, megaspore tetrad. It's a linear tetrad. Okay, just understand, megaspore mother cell. This cell first forms a dyad here, then forms a tetrad here. Okay, in the same place of the nucellus. So this is after meiosis 1 and this is after meiosis 2. Okay, so now what is formed? A megaspore tetrad is formed. So what will be here? Megaspore tetrad will be here. Then understand, this is called the megasporogenesis, formation of megaspores. Okay, then understand, in this further development, most commonly the upper 3 cells degenerate and only the lower 1 cell become functional. This is called the functional megaspore. This is the only megaspore functional in most of the cases. So, the formation of megaspore from megaspore mother cell by meiosis is called megasporogenesis. Megasporogenesis is complete, and as a result of megasporogenesis, what has formed here? Single megaspore has formed, the other 3 degenerated. Do you get me? So now, during the developmental stage, now the nucellus at the micropylar end, 1 megaspore mother cell undergo meiosis to form 1 megaspore, 1 functional megaspore normally. Okay, and this is now not the mother cell, it is now the functional megaspore with the haploid content. Okay, then what happens to this? What are the further changes? Earlier, microspore has modified to pollen grain. Microspore formed like this, all the 4 are formed. all are dissociated to pollen grains. But here again some more developmental stages are there. Okay, so understand this functional megaspore undergoes the further development, and that development is called monosporic development. Okay, monosporic, single spore is getting developed. Okay, in monosporic development, what happens is the single functional megaspore, this is not the megaspore mother cell, this is a megaspore. Okay, its nucleus only divides to form a two nucleated cell. Okay, first it forms two nucleated cell. Okay, and again its nucleus undergo repeated division to form 4 nucleated, 8 nucleated cells like this. Okay, single cell will be having 4 nucleus. Okay, here again it undergo repeated nuclear division, means it become 8 nucleated. So we can call this all, it is forming 2, 4, 8 nucleated structures. These structures we are calling as embryo sac. So the megaspore develops to embryo sac. Embryo sac is equivalent to pollen grain. It is the female gametophyte. Pollen grain is the male gamete carrying structure. This will be the female gamete carrying structure. So female gametophyte. Okay, so understand, its nucleus undergo repeated mitotic divisions to form 2 nucleated, 4 nucleated, 8 nucleated embryo sac. Then in the 8 nucleated embryo sac, there is some rearrangement of the nuclei. I am drawing it better here. Okay, in the 8 nucleated embryo sac, one each nucleus from the poles migrate to center. Okay, 3 each remains at the pole. One each nucleus from this will be moving towards the center. And later, this deposits the cell wall. Okay, cell wall will be deposited, and here will be 3 cells at one end, 3 cells at the other end. Okay, here 3 cells, and there will be a central cell, big central cell with these 2 nuclei. Okay, in a mature embryo sac, the assemblage is like this. 3 nuclei at the one pole develop the cell wall around it, 3 cells at one pole, 3 cells at the opposite pole, and 2 nuclei in the center remain in a large cell. Okay, then we can just label this central cell. This is the central cell. Okay, it has the 2 nuclei which has come from the poles, from both the poles. So we can call them as polar nuclei. Okay, since they have come from the poles, they are called polar nuclei. Then the 3 cells at, this is the micropylar end, this is the chalazial end, means the 3 nuclei at the chalazial end are called antipodals. Okay, the 3 nuclei at the micropylar end consist of egg apparatus. This form egg apparatus, micropylar end, down version, tip portion. Micropylar end forms the egg apparatus. Then at the chalazial end it forms antipodal cells. Okay, 3 cells at the chalazial end are called antipodals. 3 cells at the micropylar end form egg apparatus. In the egg apparatus, middle one is the egg. Okay, the 2 surrounding ones are called synergids. The central one is egg. The 2 surrounding cells are synergids in the egg apparatus. This is the structure of a mature embryo sac. It is called 7-celled, 8 nucleated. Typically, mature embryo sac is 7-celled and 8 nucleated. You can see 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7. 7 cells but 8 nuclei. So, mature embryo sac is 7-celled, 8 nucleated. This type of a structure will be formed here. So, at the micropylar end, this type of a structure is formed. Okay, and it will be having 3 cells here, the egg apparatus, 3 cells here, the what? Antipodals, two nuclei and a central cell at the middle. Okay, this is what is the embryo sac. Did you get me? So what the mature ovule consists of? It consists of embryo sac inside. The embryo sac has the female gamete, egg. So we have completed the development of egg also. Last class we have discussed the development of male gamete inside the pollen grain from the generative cell. Two male gametes are formed. And here now it's clear, I think, where is the female gamete formed. Female gamete is formed within the nucellus inside the embryo sac. Okay, after megasporogenesis, one megaspore is formed. This megaspore develops to the female gametophyte embryo sac. Within the embryo sac, the female gamete egg is formed. Okay, I think it's clear. The female part gametogenesis. Megasporogenesis and monosporic development of embryo sac and the formation of egg inside the embryo sac. So I think it's clear. The remaining session we will, any doubts we clarify in the live session, and thank you.